reproduction in organisms. So reproduction is essential to form a offspring. Offsprings means it gives a birth young ones. Hence, reproduction is a divided into the two types. One is a asexual reproduction, another type is a sexual reproduction. Under these modes of reproduction is fertilization to form a new individuals. That means a sperm and ova to take part into the fertilization and to form a zygote. This zygote has been to develop various changes to form a new organisms. That is called as a reproduction. Under the reproduction, two types of reproductions. One is called as asexual reproduction. Another one is called as a sexual reproduction. Asexual is a without gametes to form a new young ones. That will be take part into the invertebrates. Now sexual reproductions, they have involved into the gametes such as into the sperm and ova. They form into the fertilization to give a young ones. That process is called as a sexual reproduction. Now asexual reproduction is a amitosis, somatic, blastogenic reproductions. In this following methods, asexual reproduction has been to take part. So amitosis is a without any cell divisions. That means a parent body itself has been to divide into the two halves, or a body itself, parent body itself, they form a extra growth. Then they are going to be developed. When it is going to be developed, they are separated from the mother body. It's called as into a asexual reproduction. So under this asexual reproduction in the living organisms, that means in different organisms based on the cell walls like protista, bacteria, archaea, it is a uniparental inheritance. So uniparental inheritance means that is not going to be forefather character has been to transfer from the one generation to another generation. The body itself has been to divided. A mother body itself of the nucleus has been to expand and to form a new young ones. That is called uniparental inheritance. Now under this asexual reproductions, various methods are going to be taken place. That is one is called as a budding method, germinal method, fission method, sporulation method, fragmentation method, and regeneration method. Now fission has been classified. So first, fission is a mother body has been divided into the two equal halves. That is called as a fission. That is why the definition is a mother body to form two identical daughter individuals are given. So fissions has been classified. That is called as sporulations method, multiple fission, binary fission, strobilation method. So fissions are four types of the fissions are takes place in the living organisms. One is called sporulations, another one is called as the multiple, the next one is a binary, then next one is a strobilations. Now, under this fission method, a fission has been further classified. One of the classify is called as a irregular binary fissions. Now, this irregular binary fissions has been to take part in the amoeba. So, amoeba is a single cell. Now, this is a amoeba where it is irregular in shape. Any region of the amoeba has been to take part into the divided. Now, that division of the part has been make them to complete body. That means a complete living organisms. That type of the fission is called as irregular binary fissions. Now you are seeing here this is the diagram. Now is one amoeba. Now they are going to be from that mother body to form irregular another form. When it forms into as like a mother body, they are divided into the two daughter cells. So here, without any exchanging of the nuclear material, the body itself has been to divide into the two daughter cells. That is called irregular binary fission in amoeba. Now let's go to the another method. That method is called as a transverse fissions. So fission, we know that fission is a divided into the two body. Now in this transverse, example is parameciums. So this paramecium is found into the marine. Now this paramecium has been to divided from the mother body. So one paramecium has divided is a transverse. So that condition forming of two paramecium, this method is called as a transverse method of fusions. Okay, now another method from the binary fissions, that is called as a longitudinal fissions. So first we saw that is a irregular, example amoeba. Second method of fission is a transverse, example is a paramecium. Then third method is called as a longitudinal method. This longitudinal method example is a euglena. Now this euglena, now you know the structure of euglena. Now this euglena having a nucleus. Okay, now having chromosomes. Okay, now from this body itself, they are going to be fission has been formed. A septa has been formed. A body has divided into the two halves. See this process. Second process, what happened? This a long tuft flagellums has been divided. Second stage. Third stages. Now first stage goes to the second stage. This chromosome or nuclei has formed into the two. From the single nuclei, they are formed into the two. Next stage, they are dividing two nuclei. Then last stage, these two nuclei separately has been divided, and body also has going to be septa, walls or forms. Once they are going to be divided, these two nuclei, then they are separated and to form a new offspring. This method is called as a longitudinal method of fissions. Example is a euglena. So once again, the longitudinal process is from the euglena, a nuclei has been divided into the two nuclei. Now, after the division of these two nuclei, a walls are formed. When they are completely divided, then the body has going to be divided. So once they are divided, it forms a new euglenas. So without any exchanging of inherited characters, or they are not exchanging of nucleic material, as it is they are formed into the two N1s. This is called as a longitudinal method. Now next method, that is called as a multiple fissions. Now this multiple fission is a process where there is going to be asexually. This will be take part into the reproduction of malaria. That is a plasmodium is a parasite. Plasmodium is a parasite. Under this plasmodium has transferred the diseases. A number of plasmodium has been to produce asexually method. So from this is called scission. This is a scission. Now having single nuclei, various processes, finally it gives into the scission under asexual reproduction. This scission has to form, you are seeing this scission, only one nuclei, isn't it? Now next stage what will happen, a one single nuclei has to form a number of nuclei. So number of scissions, now number of nuclei means they are forms a number of daughter cells. When this number of nuclei are being to form, each this nuclei has been matured. The third stage is called as daughter cells are going to be maturing. Now when it completely a daughter cells, that is called plasmodium, has been to form, completely formed, it is a mature, that means it is ready to attack, to cause us a fever. Okay, so this is asexuals. Now when it matured, the wall has going to be ruptured. So a number of a scission rupture, the forming of a new plasmodium, that is called merozoites, a liberating of merozoite. Single nuclei has to form a number of daughter cells. Now these daughter cells are mature, they itself, when they are matured, they are going to be break down the wall of the scission and form a new cells. That cells are called as merozoites. This process is called as multiple fissions. Okay. Now, like that, another method from the fission method, that is called strobilation in Aurelia. Strobilation in Aurelia. Now, this is a strobilation. Now, this strobilation structure having a stolon, stalk, now that is a mansua, now having a mouth, now this is called as tentacles. This Aurelia has been moved from one place to another place, that is having tentacles are going to be used. Now this is a structure of Aurelia. So what will happen in asexual reproductions, now these regions, now you are seeing this like a cup shaped structure. Now these divisions, a one division, that is called as ephira. Now this ephira, each ephira has been to develop like a separate from the parent body. Ephira has going to be formed separately. So there is called as a yearly ephira. Now this yearly ephira has developed, it's like a mother body. This is called as stobilization. Further ephira has formed, it take part into the formation of mouth, then the tentacles are formed. Once it looks like a structure has going to be formed, it becomes a young ones, complete young ones. So what here has going to be explaining, there is no gamete, only the body, ephira portion itself to form a separate, then separating to develop as like a complete structure of the mother body. Okay, this is called as a stobilation process. Okay, in Aurelia. Now, next process is of fission budding methods.